So we just spent some time reviewing the neuromuscular junction and the process of excitation-contraction coupling. One way that we can piece all of that together is by comparing the action potential of a neuron, the action potential of a muscle fiber, and the mechanical contraction and relaxation cycles of a muscle fiber. And that's exactly what we're doing here on this slide. We have all three of those things on the exact same time scale. So up at the top, you're seeing the action potential that's recorded from an alpha motor neuron. The middle panel shows you a muscle action potential. And in the bottom panel, we have a twitch, which is the term that we use to describe a single contraction relaxation cycle inside an individual muscle fiber. So this is really a nice way of demonstrating that the alpha motor neuron has to fire the action potential first. That allows the acetylcholine to get released into the neuromuscular junction and the muscle fiber action potential to get initiated. If you compare those two action potentials, you can see they look incredibly similar. And they are. They're both electrical events, and they're due to many of the same channels, voltage-gated sodium, voltage-gated potassium channels. There's only a really short delay between the two action potentials. But if you look at the bottom panel, you can see that the twitch has a slightly longer delay before it enters its contraction phase. In other words, depolarization happens and then contraction happens. And during that 4 to 5 milliseconds, which is known as the latent phase, that's when the molecular events of excitation-contraction coupling are happening. So that delay really exists because the calcium has to get out of the sarcoplasmic reticulum. And that takes a little bit of time because that whole process isn't electrical anymore. You have to have those conformational changes in DHP and RYR. The calcium has to diffuse through the cytosol in order to get to troponin. And troponin has to move tropomyosin out of the way, and only then can that whole filament sliding process actually begin. So this is a nice way to summarize from this slide. We're going to spend the remainder of our time in this unit talking about muscle mechanics. So the job of a muscle at the end of the day is to generate force or tension, and it does this by contracting. It then returns to rest and decreases tension through the process of relaxation. Now, there are many things that can affect the ability of a muscle to generate tension. Some things work at the level of individual muscle fibers, and I'm going to talk about those things first. So individual skeletal muscle fibers have a resting length, and that length is optimized for maximum tension generation. You can see that in the length-tension relationship, which is the graph that we're looking at on this slide. So the tension is on the y-axis, and the resting muscle length is on the x-axis. The length-tension relationship is really describing the optimum length of a sarcomere. To generate the maximum amount of tension possible, a sarcomere should have some overlap between actin and myosin so that cross bridges can easily form when the process gets initiated. But it shouldn't have too much overlap at rest, because if it did, there wouldn't be anywhere for the filaments to actually slide to. They'd run into each other. So the sarcomere up at the very top, at the center of this graph, represents the ideal scenario. It's the optimum resting length. And on either side of that, you can see situations where sarcomeres are not at their optimum resting length. And that gives them a decreased ability to generate force, right? They're further down on the y-axis. So on the far right-hand side of the graph, we have a situation where a muscle has been stretched or extended beyond its resting length. If you take a look at the arrangement of the sarcomere, you'll see there's very little overlap between actin and myosin. So this makes it difficult for cross bridges to form and for the filaments to actually slide. And that's why the ability to generate tension in a stretch muscle is relatively low. If you take a look at the opposite extreme on the left-hand side, you'll see there a muscle that has a very short resting length. In other words, its sarcomere is already very, very, very compressed. And even though there's a lot of overlap between the thick and thin filament, there's just no room to slide the filaments. There's no room adjacent to the M line for the actin to slide over. So in this situation, muscle is also unable to generate a lot of force. Moving on, when we talked about graded potentials earlier in neurons, remember I made a point to mention how a single EPSP wasn't really enough to do anything. The same is true for a single twitch. This is the reason why drugs like succinylcholine are really problematic. In the same way that EPSPs can be temporally or spatially summed to reach threshold, the the tension generated by skeletal muscle can also be summed when it's stimulated at a really high frequency. So in this graph, you can see how a single stimulus, which is essentially a single muscle action potential in a twitch, only generates a small amount of tension, and it only lasts for a short period of time. But when you have repeated stimuli, that allows the tension to build more and more and more as you move to the right on these graphs. So in order for muscle to work properly, it needs to be able to achieve maximum tension within each muscle fiber. As the frequency of stimulation increases, the fiber will go through a tension building phase that's called unfused tetanus. And that's when the tension is building unevenly. So in other words, you can see there's a small decrease followed by an increase, and then a small decrease followed by an increase. Kind of like steps overall, you could see that on the left-hand side. But when the frequency of stimulation becomes fast enough, the graph starts to smooth out, and we're actually able to get all the way up to that maximum state of tension. And that's known as fused tetanus. Now, unfused and fused tetanus shouldn't be confused with the disease that's also called tetanus that some of you might be familiar with. That disease-based tetanus is a bad thing, but muscle tetanus is not a bad thing. In fact, it's necessary for muscle fibers to reach fused tetanus because in that state, they're generating the maximum amount of tension possible. Additionally, when they're held in that state of contraction, it allows multiple muscle fibers to work together as a unit within a single muscle group. But if muscles stay in this tetanic state for too long, then they fatigue. They become weak and they lose their ability to generate tension. And you know this already if you've ever pushed yourself really hard when you were working out. Fatigue has been classically understood through something known as the depletion theory, which basically says that the muscle fiber eventually runs out of the stuff that it needs to maintain the contraction, specifically things like oxygen and ATP. And that's why those differences between type 1 and type 2A fibers were sort of significant. We talked about those in the very first video. But this is also the reason why muscle has a protein known as creatine that can be used to store ATP when a muscle has a surplus of ATP. It can do that by adding a phosphate to the creatine protein, and that converts creatine into phosphocreatine, which then stores the energy for later use. When a muscle is working really hard, it can then go back to that phosphocreatine and remove the phosphate to get the ATP back. And this is the reason why a lot of people who work out have historically taken creatine supplements in an effort to increase the energetic capacity of their muscle. But unfortunately, there's not a lot of evidence, or there's conflicting evidence about whether or not those supplements are actually effective. And that's mostly due to bioavailability. And because in order to make phosphocreatine, you need to have excess ATP. You also need to have the enzymes needed to make that conversion to begin with. So just supplying the template might not really do anything. So what usually happens to people that take supplements is they increase the solute concentration in their tissues, and that causes their tissues to become hypertonic. Those people retain water, and so these things are often linked to weight gain. More recently, people have hypothesized that muscle fatigue involves changes to calcium cycling during excitation-contraction coupling. In fact, there's pretty good evidence that a lot of the important proteins that we've talked about in this unit, like for example troponin, become less sensitive to calcium in states of muscle fatigue. So there's actually some changes at the molecular level as well.